Welcome to PG Pathshala Biophysics, and today I will be talking about module on cell to Ecm interaction. I am Dr. Rubidhar. I am a DBT Biocare Fellow and I work at AIIMS, New Delhi. Now to start with a brief introduction about what do we mean by Ecm and what its role. Now the extracellular matrix, this is the full form of Ecm, which lies outside the cell, largely consists of protein biopolymers. It provides structural and biochemical support to the cells within a tissue. While the Ecm has long been viewed as a static home for cells, a growing body of work is revealing that the physiochemical properties such as the stiffness and structure of Ecm can drastically affect cell behaviors in ways similar to soluble biochemical signals. In this context, interactions with the Ecm regulate signaling and gene expression that underlie cellular processes during development, homeostasis, wound healing, and cancer invasion. Thus, it is very important to understand the crosstalk between the cell and the Ecm. The focus of this module is to understand the components of Ecm and various ways in which it interacts with the cell and maintains cellular homeostasis. Now, this is followed by the objectives, introduction to the Ecm and its function. Next, we will discuss about the composition of Ecm and how the Ecm crosstalks with the cell. Now, the most important protein in this category are the integrins. So, we are going to talk about integrin signaling and the new types of molecule called the matrix cellular proteins and some non-integrin proteins for cell to Ecm interaction. Now, starting with the introduction about Ecm. Now, the extracellular matrix is the non-cellular component present within all tissues and organs and provides not only essential physical scaffolding for the cellular constituents, but also initiates crucial biochemical and biomechanical cues that are required for tissue morphogenesis, differentiation, and homeostasis. The Ecm is composed of a large collection of biochemically distinct components including proteins, glycoproteins, proteoglycans, and polysaccharides with different physical and biochemical properties. The Ecm of each tissue has a unique composition and topology that is generated during tissue development through a dynamic and reciprocal biochemical and biophysical dialogue between the various cellular components. Example, epithelial, fibroblast, adipocyte, endothelial elements and the evolving cellular and protein microenvironment. Indeed, the physical, topological and biochemical composition of the Ecm is not only tissue specific but is also markedly heterogeneous. Now, cell adhesion to the Ecm is mediated by Ecm receptors such as integrins, discoidin, domain receptors or the DDRs and the syndicans. Moreover, the Ecm is a highly dynamic structure that is constantly being remodeled either enzymatically or non-enzymatically, and its molecular components are subjected to a myriad of post-translational modification. Now, functions of the Ecm. So, what does the Ecm do sitting in the cell? Ecm plays a vital role in regulating various functions at cellular and tissue level. Through its physical and biochemical characteristics, the Ecm generates the biochemical and mechanical properties of each organ such as its tensile and compressive strength and elasticity, because it's providing a structure, a stiffness to the organ or a tissue. It mediates protection by buffering action that maintains extracellular homeostasis and water retention. ECM directs essential morphological organization and physiological function by binding growth factors and interacting with cell surface receptors to elicit signal transduction and regulate gene transcription. It is also involved in various remodeling processes like wound healing, angiogenesis, cell motility, migration, and thus plays a key role in cancer cell invasion, because it is again cancer cell invasion is related to motility and the movement of the cell migration. The importance of the ECM is vividly illustrated by the wide range of syndromes, which can be anything from minor to severe, that arise from genetic abnormalities in the ECM proteins. Now, the next step is to understand the composition of ECM. The ECM is composed of two main classes of macromolecules. One is the proteoglycans and second is the fibrous proteins. Now, elaborating on the proteoglycans, these are proteins substituted with glycosaminoglycans, which we also call them GAGs, GAG, that generally either decorate the cell membrane or fill the extracellular space. GAGs are linear polysaccharides consisting of a repeating disaccharide, generally of an acetylated amino sugar alternating with a uronic acid. Proteoglycans constitute a major component of the extracellular matrix and have been particularly well analyzed in the context of cartilage, where the matrix constitutes more than 90% of the dry weight of the tissue. Although there are intracellular types, most proteoglycans are destined for the extracellular space. These are very diverse molecules, and various combinations of both different types of proteins, whether it's matrix, cell surface transmembrane, or covalently linked to membrane glycosyl phosphatidyl inositol GPI, and the classes of GAGs chains like the hyaluronan HA or the chondroitin sulfate, which we call them in short CS, the keratin sulfate KS, and the dermatin sulfate and the heparin sulfate are found in vertebrates. Now, for example, syndicant transmembrane proteins can be decorated with 2 HS and 1 CS chain. Aggregant cartilage matrix protein is typically decorated by about 100 chondroitin sulfate and keratin sulfate chains, so it varies. Decorin, a major proteoglycan of the interstitial matrix, contains only single DS chain. Now, function of proteoglycans. Two major families of cell surface, HSPGs, syndicans and the glypicans have been identified, which bind a multitude of growth factors and extracellular matrix molecules and have been implicated in several signal transduction pathways that regulate cell proliferation and cell shape. While syndicans are transmembrane proteins with an intracellular cytoplasmic domain, glypicans are attached to discrete cell membrane regions by a GPI lipid anchor. Proteoglycans have a wide variety of functions that reflect their unique buffering, hydration binding and force resistance properties. For example, in the kidney glomerular, perlecan has a role in the glomerular filtration. By contrast, in ductal epithelial tissue, decorin, biglycan and the lumican associate with collagen fibers to generate a molecular structure within the ECM that is essential for mechanical buffering and hydration, and that by binding various growth factors provides an easy enzymatically accessible repository of these growth factors. So, there are various functions of the proteoglycans as we discussed. The next important component of the ECM are the fibrous proteins. The main fibrous ECM proteins are collagen, elastin, fibronectins and the laminates. Starting with the collagen, collagen is the most abundant fibrous protein within the interstitial ECM and constitutes up to 30% of the total protein mass of a multicellular animal. Collagens, which constitute the main structural element of the ECM, provide tensile strength, regulate cell adhesion, support chemotaxis and migration and direct tissue development. Principal producers of collagen fibers are fibroblasts. Epithelial and smooth muscle cells also secrete their own type 4 collagen. Now, next is the elastin. Elastin fibers provide recoil to tissues that undergo repeated stretch. Importantly, elastin stretch is crucially limited by tight association with collagen fibrils. Elastin is found in artery walls, in the lungs, in the intestines and of course in the skin. The next group of proteins in the fibrous protein category are the fibronectin. A third fibrous protein, fibronectin, is intimately involved in directing the organization of the interstitial ECM and additionally has a crucial role in mediating cell attachment and function. Fibronectin can be stretched several times over its resting length by cellular traction forces. Such force-dependent unfolding of fibronectin exposes cryptic integrin binding sites within the molecule that results in pleiotropic changes in cellular behavior and implicate fibronectin as an extracellular mechanoregulator. Fibronectin 3 domain contains the amino acid sequence responsible for integrin binding and heparin binding domains, which are further discussed later in this module. Two other domains of fibronectin, the FN1 and FN2 domains, are smaller than the FN3 domain with 45 to 60 amino acids, but they share the basic anti-parallel beta sheet structure stabilized by disulfide bonds. The fibronectin domains are primarily responsible for the binding of fibronectin to collagen, fibrin and other fibronectin molecules and are often present in multiple copies within each protein. Now, the last in these groups are laminates. These are mainly present in the basement membranes and partly responsible for providing the tensile strength of the tissue. Laminin consists of three subunits alpha, beta and gamma, which come together to form a characteristic cross pattern that can bind to other laminins as well as proteoglycans and other ECM proteins. So, now we understood the components of ECM and next we will try to understand the crosstalk between ECM and the cell. Cells respond to the mechanical and biochemical changes in ECM through the crosstalk between integrins and the actin cytoskeleton. What are integrins? So, integrins are heterodimeric transmembrane receptors composed of 18 alpha and 8 beta subunits that can be non-covalently assembled into 24 different combinations. Integrins have unique ability to respond to the molecular composition and physical properties of the ECM and integrate both mechanical and chemical signals through direct association with the cytoskeleton, which also determines the selection of specific integrin species to be involved. So, depending on the function, which combination of integrin plays a role. Integrin recognizes and binds to RGD motif, that is arginine, glycine and aspartate, which was first discovered in fibronectin, but later was found in many other Ecm proteins including laminin, tenascin, vitronectin and thrombosed bonding. Both the alpha and beta subunits of integrins bind to the RGD sequences, and the specificity of integrin binding to different matrix proteins is determined in part by other amino acids surrounding the RGD sequence. Now, the mode of action of integrins, like how actually they function, the signaling. So, there are different modes. So, one is the focal adhesion. The scaffolding interactions of integrins to the actin cytoskeleton involve actin polymerization and actin linking molecules whose interactions regulate and are regulated by the associated adhesion signaling molecules. When inactive, integrins are unable to bind to Ecm or other proteins, which is important for circulating cells such as lymphocytes. The initial assembly of the nascent adhesion is mediated by anchoring proteins called tallins, whose amino terminus interacts with the cytoplasmic tail of the integrin subunit, while the carboxyl terminal of tallins bind the actin. However, to achieve maximal integrin activation, the cooperation of another anchoring protein called kindlin, which binds to the integrin tail distinct from those used by tallin, is required. So, the synergistic effect of tallin and kindlin on the activation and assembly of adhesion structures is enhanced by the binding of vinculin to tallin, which triggers the clustering of activated integrins, actin complexes further strengthening the focal adhesion interaction. The progression from the rather unstable nascent adhesion to dot-like focal complexes upon recruitment of vinculin leads to the formation of larger focal adhesion assembly and the conformational transition of the entire complex. Integrin dimers to their active state, capable of high affinity interactions with Ecm ligands, eventually lead to maturation of fully activated streak-like fibrillary focal adhesion, which can transmit the signal. Now, another mode of action of integrin is the integrin adhesome. The integrin adhesome consists of up to 156 distinct components, most of which are a fundamental part of the adhesion sites, while others may only transiently associate with the adhesion site to influence the structure or signaling activity. As the cytoplasmic domain of integrins have non-catalytic activity of their own, they must recruit accessory molecules which contribute to the actin cytoskeletal reorganization and endow catalytic activity to the focal adhesion. The integrin adhesome associated proteins include a diverse range of kinases, phosphatases and adapter proteins which contribute and initiate cascades of signaling events. Several tyrosine kinases and phosphatases are shown to be localized to focal adhesions, which are necessary for the regulation of mechanosensory activity of focal adhesions. These include the focal adhesion kinases, or we call them FAK, SRC receptor type tyrosine protein phosphatases, and SH2 domain containing protein tyrosine phosphatases. Now, the last mode by which integrin crosstalks with Ecm is the turnover of the focal points via interaction to the cytoskeletal proteins. Talin also binds to actin and to numerous cytoskeletal and signaling proteins, thereby linking activated integrins directly to signaling and cytoskeletal systems. Now, there are several inhibitors of integrin activation also. Several proteins can inhibit integrin activation by competing with talin for binding to the cytoplasmic tail. Structural analysis have revealed an overall competition between talin and the filamin binding sites of integrin tail, and competition for the cytoplasmic tail binding can regulate the integrin activation. Integrins also bind to many PTB domain containing proteins including DOC1 and integrin cytoplasmic domain associated protein called ICAP1, and these can compete with talin for binding to integrin and so can impair activation. Now next, how integrin signaling happens. Now, one of the first integrin signaling molecules to be identified was focal adhesion kinase, as we discussed earlier, the FAK, which acts as a phosphorylation regulated signaling scaffold and is important for adhesion turnover, Rho family GTPase activation, cell migration, and crosstalk between growth factor signaling and integrins. Now, the second mode of integrin signaling is through SRC family protein tyrosine kinase. SFKs are rapidly activated following integrin ligand interaction, and SFK activity contributes to reinforcement of initial integrin mediated adhesions by activating downstream kinases and adapters. The third mode is the integrin linked kinase, ILK, is another key node in integrin signaling. ILK forms a heterodimeric complex with the limb domain protein called pinch and the actin and paxillin binding protein parvin. Fourth is the involvement of protein called paxillin, is another essential signaling scaffold that is recruited early to integrin adhesions. Now, fifth is vinculin, is important for integrin mediated cell adhesion, and vinculin null cells exhibit reduced spreading, enhanced focal adhesion turnover, and increased random migration. Now, apart from integrins, there are another group of proteins discovered called the matricellular proteins. Paul Bornstein turned the phrase matricellular proteins defined as secreted macromolecules that interact with cell surface receptors, extracellular matrix, growth factors, and or proteases, but do not in themselves subserve strictly or exclusively structural role. So, originally limited to spark, other proteins have been discovered called thrombosed bonding and tenacin, and this group now includes other proteins like and proteoglycans like TSP2, tenacin X, syndicans, osteopontin. Matricellular proteins, or the families to which they belong, are structurally unrelated glycoproteins that under certain conditions disrupt cell matrix interactions. So, this is counter or de-adhesion and are associated with cells and processes undergoing rapid change. Processes such as wound healing, angiogenesis, bone remodeling, reinnervation, and embryonic morphogenesis are characterized in part by cell matrix interactions that are continually changing to facilitate tissue growth and repair. Thus, a striking coincidence of expression of these proteins has been noted in osteogenic differentiation and remodeling basement membrane. Now, just to give in details about these proteins, the matricellular proteins, we start with spark. Spark, also termed as the osteonectin, is an acidic cysteine rich component of the extracellular milieu that display a high level of interspecies sequence conservation. High levels of SPARC mRNA and protein were found in somites, limb birds, and invasive cells of extraembryonic tissue, whereas SPARC in the adult was limited to tissues exhibiting high rates of cellular proliferation and remodeling, as well as to non-dividing steroidogenic cells. The temporal and spatial restriction of SPARC to certain active cellular populations raised the possibility that the protein modulated cell proliferation and acquisition of a differentiated phenotype. SPARC acts as an inhibitor of cell spreading in vitro and thus participate in cell matrix interaction during remodeling, development or wound healing. So, experiments have shown that exogenous expression of SPARC to various cell types like endothelial cells, smooth muscle cells and fibroblasts inhibited their ability of cell spreading when grown in collagen coated surface. Cell lines that overexpressed SPARC were rounded and clumped. Thus, this points to the fact that SPARC interacted with proteins at the cell surface to facilitate changes in cell shape and release of cells from their ECM. SPARC might affect cells by retarding progression through the cell cycle or by maintaining a rounded phenotype in cells that have already divided. And the next protein coming in this group is the tenascin. So, tenascin is a large multi-unit glycoprotein that include EGF-like repeats and integrin specific cell binding sequence and the calcium binding domain and possess both adhesive and anti-adhesive properties. Addition of soluble tenascin to cultured tumor cells resulted in partial detachment and loss of intercellular contacts as well as inhibition of cell migration and spreading on basal lamina. Experiments with tenascin coated substrates has shown that tenascin retards the attachment or spreading of several cell types on fibronectin, laminin and basal lamina coated plates or surfaces. Other assays provided evidence for adhesive domains in tenascin that interacted with a chondroitin sulfate, proteoglycan or with an RGD sensitive integrin. Various research shows functional antagonism between tenascin and fibronectin. The third protein in this group is the thrombosed bonding, which participates in the aggregation of activated platelets but also functions as a secreted product of most mesenchymal and epithelial cells. The protein contains globular N and C terminal domains which have been implicated in binding to heparin and to calcium and platelets respectively. It also has a central stalk-like region which interact with most ECM proteins. Integrins, heparin sulfate and so TSP's role as adhesive versus anti-adhesive protein keeps varying depending on the cell type. It promotes cell to ECM interaction in most cell types like keratinocytes, melanoma cells and platelets. On the other hand, it reduces focal adhesion in endothelial cells. And next we are going to discuss about the non-integrin proteins which are involved in cell to ECM interaction. And in this group there are two classical ones, the discoidin domain receptor, we also call them as DDRs, have recently emerged as non-integrin type receptors for collagen. The two mammalian gene products discoidin DD1 and DDR2 constitute a subfamily of tyrosine kinase receptors that are selectively expressed in a number of different cell types and organs. Upon collagen activation, it gets phosphorylated at the tyrosine residue and thus activate various downstream signaling and thus can regulate cell adhesion, proliferation, and extracellular matrix remodeling. In addition to mediating direct collagen dependent signaling, DDRs can also modulate signaling pathways initiated by other matrix receptors, example integrins, various cytokines like TGF-beta, and transmembrane receptors like Notch1 and the insulin receptor. Now, the next protein in these groups are the syndicans. Syndicans are transmembrane heparin sulfate proteoglycans that are present on most cell types. Now, these have been known for some time to regulate a variety of biological processes ranging from coagulation cascades, growth factor signaling, lipase binding and activity, cell adhesion to Ecm and subsequent cytoskeletal organization to infection of cells with microorganisms. Now, in terms of common function, all syndicans have a common carboxyl terminal F5A sequence that can interact with the PDZ domain containing proteins, which implies a role in protein-protein interaction. To date, 3 PDZ domain proteins have been shown to interact with syndicans, and these are the syntinin, the Cascade or the Lin family, and the synectins. So, with this we have almost reached the end of this module, and just to summarize the module, the Ecm is composed of a large collection of biochemically distinct components including proteins, glycoproteins, proteoglycans and polysaccharides with different physical and biochemical properties. Interactions with the Ecm not only provides the structure and stiffness to the cell, but also regulates signaling and gene expression that underlie cellular processes during development, homeostasis, wound healing, cancer invasion. Now, as we discussed that the Ecm is composed of two main classes of macromolecules, proteoglycans and the fibrous protein. Proteoglycans are proteins substituted with the glycosaminoglycans or GAGs that generally either decorate the cell membrane or fill the extracellular space. On the other hand, the fibrous protein consists of collagen, fibronectin, elastin and laminin. Major interaction between cell and Ecm takes place through the crosstalk between integrins and actin cytoskeleton. Integrins are heterodimeric transmembrane receptors composed of various alpha and beta subunits that can be non-covalently assembled into 24 combinations. Integrin recognizes and binds to the RGD motif. Talins and kindlin are the two major proteins required for integrin action. So, they generally activate the integrin signaling pathway. Integrins are inhibited by filamin and the PTB domain containing proteins including DOC1 and integrin cytoplasmic domain associated protein 1 or the ICAP1. Integrin signaling involves various downstream mediators like the focal adhesion kinase and the integrin-like kinase, the SRC family kinase, paxillin and vinculin. Beside integrins, a new class of proteins called the matricellular proteins have been discovered. They are defined as secreted macromolecules that interact with cell surface receptors or the Ecm, growth factors and proteases, but do not in themselves are exclusively play structural roles. Originally limited to spark, now there are other matricellular proteins called the tenascin and thrombosed bonding. And next we move on to the various other discoidin and syndicans, which are the non-integrin proteins involved in the cell to Ecm interaction. And thank you so much for listening to this module on cell to ECM interaction. Thanks.